Okay, good morning and good afternoon, everyone. Welcome to Indoor Ag Science Cafe March. This is a special day because we have three excellent speakers, future of the agriculture, indoor agriculture. So I'm so excited. I'm a professor at the University, Ohio State University, Cherry Kubota, and I'm the host today for this Indoor Ag Science Cafe. So we are doing this monthly, as you know, and then inviting speakers from different, the disciplines, industry, academia. So today we have three Spartans from Michigan State University. And then I'm going to introduce their titles and names in a minute. And then April cafe is going to be international. So we have to shift 1 hour, earlier than usual 11 a.m. time slot because the speaker is from University of Tokyo, Ryo Matsuda, talking about pharmaceutical production using plant grown under controlled environment. And then May, we have Rob Eddy from Resource Innovation Institute. He's going to talk about workforce development program they are launching at RII. And then June, we have another faculty presentation from US, Chris Curry, who is in Iowa State University, talking about growing coronary herbs under controlled environment. So good stuff. And then after that, July, we usually don't have any cafe, and August, we have already presenter lineup. So it's going to be good series again next academic year. So, okay, with that, quickly, we have three presenters from Michigan State University. This is Michigan State University OptimIA Research Showcase. So the first presenter is Nathan Kelly. He's going to talk about dependence on far-red light on other wave bands at increasing growth of lettuce. The second speaker is Devin Brewer, who is going to talk about improving red leaf lettuce color with endo production cooling. And then the last speaker, the third one is Jiyong Shin, and then he's going to talk about interactions between light quality and temperature in vertical farming. So the great ones, graduate students showcase. So with that, I'm going to stop sharing.
All right. Well, hi, everyone. My name is Nathan, and I'm a PhD student at Michigan State. And today I wanted to briefly discuss a study we did on far red light titled The Dependence of Far Red Light on Other Wavelengths at Increasing Growth. But first, I wanted to give a bit of background information on light and specifically far red light. So as we know, different colors of light make up different portions of the light spectrum. PAR, or photosynthetically active radiation, has been traditionally defined as light with wavelengths between 400 and 700 nanometers. And light within PAR is responsible for the majority of plant growth and morphological changes, and are the predominant wavelengths that drive plant photosynthesis. Although there has been some talk of late about the utility of far red light and specifically its impacts on driving photosynthesis, and if it should be included in PAR. But as of now, adding PAR red to the spectrum just expands PAR to a new definition called EPAR, or extended PAR. BAR, on the other hand, is a more broad range of light that includes UV and far red light due to their ability to drive biological processes. But for this study, we wanted to focus on the primary effects of far red light on lettuce. Far red light has been shown to increase growth, primarily by increasing leaf area and stem length, and this leads to a greater canopy size, which allows for more light capture and use for photosynthesis and growth. Far red light has also been shown to increase growth by stimulating photosynthesis directly. But far red light alone cannot drive photosynthesis too well, which is in part due to the red drop phenomenon, where quantum yields of far red light begin to drop off quickly after about 680 nanometers. But when far red light is paired with red light, for instance, it can lead to a greater quantum yield than the two wavelengths combined. This indicates that other wave bands are likely needed for far red light to efficiently drive photosynthesis, and that the two enhance each other. So one question we try to answer here is this effect dependent on far red light, or can far red light drive photosynthesis when paired with other PAR wavelengths, such as green light instead of red. So on to our study. Our objectives were to see if far-red light is as effective as wavelengths within PAR at increasing biomass, and to find out how the promotion and growth from far-red light depends on other light wavelengths. We hypothesized that the inclusion of far-red light in the light spectrum would increase biomass due to an increase in leaf area and by increasing the photosynthetic efficiencies of shorter wavelength light, and that in the absence of red light, far-red light would have less of an effect on plant biomass accumulation. Now, and the study was conducted in the Controlled Environment Lighting Lab at Michigan State, and this is a temperature-controlled growth chamber. Here we have 8 hydroponic racks that have a recirculating solution, and we used custom LEDs that allowed us to change the photoperiod, light intensity, and light spectrum using 5 different colors of LEDs and 2 white LEDs. And we grew a red leaf cultivar Ruxi and green leaf cultivar Rex and germinated them in Rockwell sheets at 73 degrees under a 24-hour photoperiod and a TPFD, or total photon flux density, of 180 micromoles. Then on day 10, we transplanted the seedlings into the hydroponic system and cell, where they were grown under 12 lighting treatments at a 20-hour photoperiod. Now here is an example spectrum of our lighting treatments that we used. Each spectrum had a different combination of blue, green, red, and far-red light. And here's just a spectrum showing all the peaks of each of our LEDs that we had. And each treatment had a TPFD of 176 micromoles during this time. Then here are the 12 specific treatments. I'm not going to go through each one individually, but we had a constant blue PFD of 22 micromoles in each treatment, and then 3 different green PFDs of 0, 44, or 88 micromoles. And then within each green PFD, we added green light to replace red light, and then also substituted in far red light for red light to see how an increasing far red PFD would affect growth when red light also is decreasing in the light spectrum. So in other words, we wanted to see, can far red light drive growth as far as it is replaced by PAR wavelengths, in this case, green light. Down to some brief results of this study. Here are some graphs of lettuce Ruxi fresh mass, dry mass, leaf area, and specific leaf area, which is the amount of leaf area per gram of dry mass. Each colored line we have here is a different far red PFD of 0, 22, 44, or 66 micromoles. And then we have an increasing green PFD on the x-axis. And then as a quick reminder, as we increased either the green or far red PFD, red light also decreased in the spectrum to maintain the constant TPFD of 176 micromoles. So for example, at a green PFD of 88 and a far red PFD of 66, red light was fully removed from the spectrum. So we found that shoot fresh mass was primarily affected by the far red PFD, although increasing the green PFD did slightly decrease fresh mass. Interestingly, continually adding more far red light did not increase fresh mass. In fact, lettuce grown under 66 micromoles of far red light had less fresh mass than those grown under 22 or 44 micromoles of far red light. Although for this cultivar, all far red light treatments did lead to greater shoot fresh mass than when no far red light was applied to the light spectrum. And there were a few trends with shoot dry mass, but in general, a moderate or high PFD of far red light plus no or the max green PFD led to the greatest shoot fresh mass. And we can see with leaf area that the trends are much more consistent. Increasing the green PFD slightly increased leaf area, but increasing the far red PFD had the largest effect on leaf area. Different than fresh mass, the max PFD of far red light, 66 micromoles, did lead to the greatest leaf area in this case, which indicates to us that increasing the far red PFD increased leaf area, but it did not necessarily continually increase fresh mass here. And specific leaf area followed similar trends to leaf area, but there was a greater effect of green light. The max PFD of green light plus the max far red PFD led to the greatest SLA, which means the plants had the least dense leaves, and that the increase in leaf area led to the smallest increases in dry mass. Here are some pictures of plants from each treatment. You can really see how plant shape and size change here with the addition of far red light especially, but also slightly with green light. When no far red light or green light was applied, the plants were much more compact and circular here. And as we started to add more far red light or more green light, the plants became a bit wider, more yellow, had larger separated leaves. And then we can also see the big differences in leaf coloration here. At a lower green photon flux density, far red light had less of an effect on leaf redness in this case, but at the max green and far red PFD, leaves are much more yellow than with no far red or green light. And this also ties in with our chlorophyll content measurements, where we can see the plants down here are a bit more yellow versus the ones up here that are a bit more green along with the red coloration. Then I have some results for our green leaf cultivar Rex. The figures are the same concept as the ones before. So once again, we saw that increase in the far red PFD to 22 or 44 micromoles led to the greatest fresh mass, while applying the max far red PFD or no far red light led to equally low fresh mass. This time, dry mass was more associated with fresh mass. A moderate far red PFD increased it the most compared to no or the max far red PFD. Also, increasing the green PFD to the max of 88 micromoles generally decreased dry mass compared to no green light. Then leaf area increased when far red light of any PFD was applied, but there was less separation between far red light treatments, except when there was no green light in the spectrum. This indicates that Rex may need less far red light to promote leaf expansion than Ruxi, as 22 micromoles promoted the same leaf area response as 66 micromoles. And here are the pictures of those Rex plants. Once again, you can see how leaf expansion begins to increase quite a bit as we add far red or green light. Specifically, leaf length tends to be more noticeable here, and the leaves begin to separate from each other more compared to the more compact plants under no far red or green light. You can also see the plants are a bit more yellow when we apply more far red and green light, similar to the previous cultivar. So in conclusion, far red light up to 66 micromoles increased leaf area of both cultivars, but only 22 micromoles was needed for Rex. Far red light up to 44 micromoles increased fresh mass of both cultivars, but 66 micromoles of far red light did decrease fresh mass. And far red light did decrease leaf redness and SPAD, or chlorophyll content, to some extent. Then finally, increasing the green PFD slightly decreased fresh mass, but increased leaf area of both cultivars. So in general, far red light applied without any red light with the max green PFD increased leaf area, but not fresh mass of both cultivars. This indicates that far red light only increased growth when red light was present in the light spectrum, and that other PAR wavelengths like green light may not be effective substitutes. So in general, far red light can increase yields, but generally decreases plant quality, at least at this TPFD, which is relatively low. So I think more work is needed to find out the optimal far red PFD or percentage that optimizes growth without decreasing quality attributes too much. And I'd like to thank our funders of this research, and I believe I can take any questions you may have now.
Yes, so we have 5 minutes. Oh, thank you so much.
Yeah, of course.
So we have 5 minutes, quick question and answer time. And then at the end, I think we have additional sort of panel discussion type of all together. But anyway, so I see questions from Ed. Ed, do you have microphone? If so, you might want to ask those questions.
I'll pull my slides back up too in case anyone wants to reference those.
Yeah. Ed, do you have a microphone? Otherwise, I just read this. So the first question is, what purpose does light serve during seed germination? I don't remember. Did you put light during seed germination?
Yeah, seeds were germinated under a 24-hour photo period just with warm white light.
Any thought behind that? Do you need light for germination, or you don't need light, or what is that?
In general, with lettuce, we've always used this specific germination treatments because it's been relatively consistent for us.
Yeah,
it's worked well so far. So
yeah, we tend to do the light germination too. Okay, in addition to leaf mass, other production parameters such as taste, aesthetics, aesthetics, etc. Sorry about.
Yeah, so we did not look at taste for this study. This one was conducted during the pandemic, so it was hard to like coordinate one of those on campus, even if we wanted to. But quality-wise, just from looking at the plants and handling them, when we applied a lot more far red or green light, the plants were very floppy. They could not hold themselves up on their own when we harvested them, and the leaves just felt a lot softer and thinner, and overall lower quality plant that most people would not want to buy, in my opinion.
Yeah,
we did not do any quantitative measurements of those attributes besides looking at leaf coloration and stuff like that.
Right. So everyone who have questions, you actually technically you can put your questions either in the chat or the Q&A. Q&A, we can't see the question until it is answered. So just keep in mind how it works. This is a new function for us too. But anyway, so the third question, Ed, does 88 days represent growth to maturity? Was that 88 days, your experiment? I don't think,
we grew them to 20.
Yeah, 28,
28. Yeah.
28, 4 weeks.
Yeah, we choose in general around 4 weeks because at least for our setup, that's when the plant canopy begins to close and we don't want other plants influencing the growth of others. So yeah, and you can see in these pictures here, which were taken the day of harvest, that the canopy is very much closed. Leaves have begun to already shoot up. So 28 days is about the max that we can grow in our system if we want the plants to be individual. Right.
Okay, so in the chat, more of a general question, would you say based on the presented study or other researchers, is light turnability something you envision being a big part in commercial vertical farming?
So you mean changing the light spectrum more so?
Yeah.
I think it depends more so on the intensity of light that people are using. Obviously, here we used a pretty low intensity compared to what some growers may use. And that's in part due to our specific lights we have, but then also we see more changes based on light quality at a lower intensity of light. So I would assume for this study, for instance, as you increase the intensity of all wave bands at the same amount, the responses may be less extreme, and that higher light intensity may contribute more to plant quality and yields than a specific light spectrum. But there hasn't been too much research that I've seen that are growing plants at those really high intensities of like 600 plus micromoles that are also changing light quality. So I'd be interested in learning more about that and finding studies like that at some point.
Okay, great. Well, thank you, Nathan. I need to move on to the next speaker and we come back, again, thank you very much. So the next speaker is Devin.
All right, everything looking good?
Yes, thank you.
Great. Hi, everybody. My name is Devin Brewer, and I'm here to share some information on our project, improving red leaf lettuce color with end-of-production cooling. So for some background information first, the demand for leafy greens is increasing, and this is mainly driven by its health properties, which include a large supply of vitamins, minerals, and phenolic compounds. One such phenolic compound is anthocyanins, and these are compounds associated with the red and purple coloration in the foliage of plants. And it's suggested that greater consumption of plants containing anthocyanins can reduce the occurrence and severity of chronic illnesses such as chronic inflammation, cancer, and neurodegenerative diseases. So some past research at Michigan State, we found that we could produce the greatest lettuce yield under these conditions: a day and night temperature of 28 and 21 degrees Celsius for an average daily temperature of 26 Celsius, CO2 supplementation of 800 micromoles per mole, a radiation intensity of 300 micromoles per meter squared per second for 17 hours a day, and a VPD of 1.1 kilopascals. And if you look at the upper graph on the right, you can see that as the average daily temperature increased from 20 degrees Celsius to 26 degrees Celsius, we saw a 35% increase in fresh mass. And then if you look at the lower graph, you can see that as CO2 increased from just above ambient levels at 400 micromoles per mole to 800 micromoles per mole, we saw, sorry, my screen's blocking my number, 30% increase in fresh mass. But if we increase beyond that to 1200 micromoles per mole, we actually see a 10% reduction. However, there were issues under these conditions. Red leaf varieties contain reduced anthocyanin and therefore weren't very red. And then tip burn incidence was very high. So the objective of our study was to quantify how end of production blue and red sole source lighting and cooling temperature influence biomass, nutrition, leaf coloration, anthocyanin accumulation, yield, and plant quality. Color is very important as far as consumer preference goes. It's the first thing that someone sees really when they're looking in a supermarket at your produce. So when we're talking about lettuce and color, the intensity is very important as well as the distribution of the color throughout the leaf. And when we're evaluating color, we like to use a couple different parameters. Chroma, which is the saturation or how vivid a color looks to a person's eye. A chroma of 0 would probably be pretty gray versus like a chroma of 20 is going to be more vibrant. Hue angle, which is what determines the actual color and is really the angle around this graphic. You can see at a 0 degree angle, you have red. At 90, you have yellow. At 180, you have green. 270, you have blue. And then if you go all the way back around, you're at 0 or 360 degrees, you're at red again. So this is the slide that I was on before we had technical difficulties. But in this graphic, you can see the hue angle as it moves around the circle. 90 degrees is yellow, 180 degrees is green, 270 degrees is blue, and then 0 is red. And then lastly, we look at lightness or L star with 100 being white and 0 being black. And then just to visualize this a little bit better, here we have an example. The lettuce plant on top has a hue angle of 118.5, a chroma of 28.6, and lightness of 44.9, while the lettuce plant, the lower lettuce plant has a hue angle of 40.6, a chroma of 6.4, and a lightness of 31. So you can kind of see how all of these values affect the appearance of the lettuce plant. So the cultivars we looked at for this study were a red oak leaf, Rixi, a Salnova butterhead, Barlock, and a red romaine Coues therinus. And during our growth phase, which was from days 12 to 24-30, dependent on cultivar, plants were split between 3 walk-in growth chambers or 6 deep water culture hydroponic tanks. Here's our fertility. And then we maintain pH and EC daily at 5.6 and 1.7. We maintained that temperature of 28 or 21 degrees Celsius day and night, a radiation intensity of 300 micromoles per meter squared per second, CO2 supplementation at 800 micromoles per mole, a VPD of 1.1 kilopascals, and then air velocity of 0.7 meters cubed per second. Then during our end of production phase, which lasted 6 to 8 days, depending on cultivar, we reduced radiation intensity to 150 micromoles per meter squared per second, so half, and we changed the spectrum to 75% blue, 25% red radiation. And then we exposed plants to cooling temperatures of either 8, 14, or 20 degrees Celsius. So the data we collected included colorimeter measurements on days 0, 2, 4, 6, and 8, relative chlorophyll concentration, chlorophyll fluorescence, phenolic analysis, and mineral content analysis as well. And so the results we saw concerning color, by day 4 of the 8 degrees Celsius treatment, the average hue angle of Barlock decreased 10% compared to the 20 degrees Celsius treatment. And I think it's important to remember here that a lower hue angle is more red, so that's desirable that it's lower. Rook size, average hue angle, and lightness had decreased by 12 and 14% by day 4, respectively. And then likewise, the average hue angle and lightness of thoroughness had decreased 26% and 10% compared to the 20 degrees Celsius treatment, also by day 4. And so what does this look like? Well, we can see here that, the 20 degrees Celsius Barlock plant definitely is much more green than the 14 or 8. In the 14 degrees Celsius plant, we still see some green, but definitely a much more dark, more colorful plant. And then 8 degrees Celsius is almost completely red with very little green showing at all. And then we can see the same thing with Rook's Eye here. 20 degrees Celsius, a lot of green is visible, less with 14 and even less with 8. And lastly, with thoroughness, once again, it's the same sort of trend. Very easy to identify when looking at the plants side by side. And so anthocyanin concentration, when we're looking at just our treatments, we didn't really see much of a difference, and certainly not a significant difference between the 8 and 14 degrees Celsius treatments, looking at Barlock and thoroughness. And then if we go a step further and we look at the no chilling whatsoever, so an average daily temperature of 26 degrees Celsius, we can see that in the case of Rook's Eye, there wasn't a significant difference between any of our cooling treatments, but each cooling treatment was significantly had a significantly greater anthocyanin concentration than no cooling treatment. In the case of thoroughness, we can see that the no cooling treatment was significantly lower than the 20 degrees Celsius treatment, which was significantly lower than both 14 and 8. And in the case of Barlock, we can see that 20 and 26 degrees Celsius treatments were similar. There wasn't a significant difference in anthocyanin, but 14 and 8 degrees Celsius had significantly more than both of those treatments. And so probably something everyone's very interested in is fresh mass. And it behaved as one might expect under cooler temperatures. We saw a decrease in fresh mass. However, there wasn't necessarily a significant difference in fresh mass between the 14 and 8 degrees Celsius treatments for each cultivar. We also saw that leaf number was depressed under cooler temperature treatments. And so as it's a pretty linear relationship with all cultivars, as the temperature decreased, leaf number also decreased. So in conclusion, fresh mass accumulation of lettuce can be maximized under 28 and 21 degrees Celsius, a ppFD of 300 micromoles per meter squared per second, and a CO2 concentration of 800 micromoles per mole. And tip burn was successfully mitigated with increased air movement and VPD control. We had no incidents of tip burn once we got those two parameters under control. However, anthocyanins and coloration were reduced under these higher temperatures. But the end of production cooling treatment resulted in improved coloration and improved anthocyanin accumulation. Fresh mass and leaf unfolding was negatively impacted by the lower average daily temperature. However, I think it's very important to note that the duration of our chilling treatments was 6 to 8 days. And across the board, we saw that the majority of coloration had occurred within the first 48 to 96 hours or 2 to 4 days. With all that said, I'd like to acknowledge everyone who makes our research possible. Definitely acknowledge OptimIA. And I can take any questions at this time.
All right. Well, thank you so much.
Yeah,
Nice presentation. Okay. So any questions out there? Let's see. Great talk. How did you all decide to use HCl color scale versus other color scales such as LAB or RGB?
We had just, when looking at other papers, it seemed that HCl gave a better picture and was easier to understand than LAB, which we took measurements in LAB and converted them to HCl as well.
Okay, great. So I have a question, Devin. It was quite interesting temperature approach.
Yeah,
So I believe you reduce the light also or no?
yes.
What's the thought behind? Yeah, maybe you can clarify a little bit. Why you have to reduce the light?
So with my first project, we reduced the light and we were looking at different ratios of red to blue. So 100, 0, blue red, 75, 25, blue red, and 50, 50, blue red. And originally we reduced the light just to see what would happen and maybe find a more sustainable approach and see if it would make a large difference in coloration. We found that it did negatively affect coloration to reduce light and anthocyanin content, of course. However, we were also able to determine that light intensity had more of an effect than light quality, which I think is important. And then when you combine that with a cooling treatment, the coloration and anthocyanin content is actually improved still.
So it's to maximize the response, you reduce the light and reduce the temperature.
Right, right.
Okay.
Reducing the light might also offset some of the costs of reducing the temperature.
Ah, I see. You did a economic analysis for this?
We haven't done the economic analysis on my research yet, but we have on Sean's.
Okay, great. All right. Which one should I pick? Just the order. Did you look at impact on post-harvest quality?
What all would you say is included in post-harvest quality? We looked at mineral nutrients and different phenolic content.
Yeah, but how long is it, the shelf life, for example?
Okay. We haven't looked at shelf life as far as red leaf lettuce is concerned. We're planning to do a study in the fall, though, that looks at microgreens and some of these same parameters and end of production treatments. And we're going to look at shelf life when we look at microgreens.
Okay, great. Let's see. Energy penalties quantified for increased air movement and then for cooling. I guess this is we discussed later because that's not really you didn't do any energy analysis for this research, right?
Yeah, not yet, at least.
Yeah. Okay, and then let's see. I have a question regarding color hue measurement and leaf pigmentation. Can I ask with the microphone? Oh yeah, sorry.
Yes, please.
Yeah, that's Hans. Okay, you go ahead.
Hi, everyone. And yeah, thank you, Devin, for the presentation so far. So yeah, I have a question around the methodology of color hue measurement and pigmentation. So 2 questions.
Okay.
Did you measure the whole plant? Like you showed the top of the plant. And did you then take the hue measurement of the whole plant or individual leaves?
So while measuring, we took one of the top canopy leaves, and I would take 3 measurements across the entire leaf and then average that. And it would be the same leaf roughly for each plant.
Got it. Okay. And then the second question is, so when you're using light to impact anthocyanin content and leaf coloration, you sometimes will see where there's shade on the leaf, maybe the coloration is not as deep or it doesn't switch to that purple. Right. How, at a qualitative level, how does chilling of the lettuce impact that coloration?
So on a qualitative level, yeah, I think I understand what you're asking. You mean like when you're just using like a more intense light that if one leaf shades another, the bottom leaf wouldn't quite get that coloration, right?
Yes, exactly.
Cooling, I think, was a much more viable way to get around this. There was still, like at the very base of the leaf, it wouldn't be colored all the way, but it was certainly, in my experience, much more, it wasn't quite such a, red to green change just by the shading. Like the red kind of faded as you went further down the leaf into the shaded area, if that makes sense. Yeah, I think, does that answer your question well enough?
Yeah, no, it's interesting. Thank you.
Yeah, no
All
problem.
right. Okay. All right. Thank you so much, Devin.
Yeah,
Okay. So the third presenter is Jiyong Shin. You can start presentation.
Let me share my screen. Can you see the slides?
Mm-hmm.
Okay, let me start. Hello, everyone. I'm Jiyong Shin at Michigan State University, and I'm going to talk about the interactions between light quality and temperature in vertical farming today. Firstly, I would like to introduce why light quality and air temperature should be fine-tuned in vertical farming. A vertical farm is a plant-growing system in an almost thermally insulated space where sunlight cannot reach. And because there is no sunlight, electric lighting is needed. Also, because it is almost thermally insulated, the temperature should be regulated by an air conditioner. And for this reason, electric lighting and temperature control accounts for the greatest operational energy load in vertical farming, along with the humidification. As mentioned in the previous presentations, light quality can change the growth and shape of a plant, especially low red to far light ratio and low blue photon flux density, which refers to low blue light intensity, can induce elongation and expansion growth of a plant, which is collectively called shade avoidance responses. And the elongation growth and expansion growth to a moderate degree can increase the overall growth of a plant. Similarly, higher air temperature also can induce elongation and expansion growth of a plant, such as a stem elongation, leaf expansion. And this can increase the growth and development as well. The similarity between the responses of plants to light quality and air temperature, such as stem elongation, leaf expansion, leaf height and reduced branching, suggests the possible existence of the interaction between them. So, due to the respective importance of light quality and air temperature, and also due to the similarity between them, we decided to study their interactions. And we hypothesized that there will be interactions between light quality and air temperature in determining the growth and quality of lattice and basals. So we treated 3 different red to far light ratios and 2 different blue photon flux densities at 2 different air temperatures. And this table shows the light intensity of our lighting treatments, and we treated blue photon flux density of 40 or 100 micromole per square meter per second, and we treated the red to far light ratios of 4:0, 4:1, and 4:2, which means more red light was replaced by far light. And the total photon flux density was the same among the treatments. And we treated 66 degrees Fahrenheit as our moderate air temperature and treated 75 degrees Fahrenheit as our higher air temperature. And CO2 concentration was maintained within the ambient level, and vapor pressure deficit was similar between the temperature treatments. And we grew red oak leaf lattice Fuchsia and basil prospera for this experiment. And the lattice and basils were harvested on day 19 and day 32 respectively. And we collected shoot fresh weight, shoot diameter, and leaf coloration data for lattice, and collected shoot fresh weight and leaf coloration data for basils. And this experiment was replicated twice. Now, let's look at the results of this experiment. And in the results, I would like to talk about the interactions between red to far red light ratio and air temperature first. And after that, we'll talk about the interaction between blue light and air temperature. So the plants in the left column are representative plants grown at red to far red light ratio of 4:0. And in here, data from different blue foot and flux density treatments were averaged. And the plant in the middle and right column was grown at red to far red light ratio of 4:1 and 4:2 respectively. And at 66 degrees Fahrenheit, and as the red light was more replaced by far red light, the shift fresh rate of lattice increased by 11%. At 75 degrees Fahrenheit, as the red light was more replaced by far red light, the shift fresh rate of lattice also increased, but to a greater extent. How about shift diameter? At 66 degrees Fahrenheit, as the red light was more partially replaced by far red light, the shift diameter increased by 19%. And at a warmer temperature, the shift diameter of lattice also increased, but to a greater extent, just like fresh rate. How about leaf coloration? At a lower temperature, the leaf redness of lattice was not statistically affected by red to far red light ratio. But at a warmer temperature, the partial replacement of red light with far red light decreased the leaf redness of lattice by 52%. Next, let's look at basils. At a lower temperature, as the red light was more replaced by far red light, the shift fresh rate increased by 42%. And at a warmer temperature, the shift fresh rate of basils also increased, but to a greater extent. How about leaf coloration? At a lower temperature, as the red light was more replaced by far red light, the leaf yellowness of basil increased by 16%. But at a warmer temperature, the leaf yellowness also increased, but to a lesser extent. So to summarize the interaction between red to far red light ratio and air temperature, the higher air temperature amplified the effects of a low red to far red light ratio on increasing lattice growth and size, but at the expense of crop quality. And the higher air temperature amplified the effects of a low red to far red light ratio on increasing shoot growth of basils, but with no effect on crop quality. Now, let's look at the interaction between blue foot and flux density and air temperature. At a lower temperature, as blue light intensity increased, the shift fresh rate of lattice decreased by 14%. At a warmer temperature, as blue light intensity increased, the shift fresh rate of lattice also decreased, but to a greater extent. How about shift diameter? At a lower air temperature, as blue light intensity increased, the shift diameter decreased by 6%. And at a warmer temperature, the shift diameter of lattice also decreased, but to a greater extent, just like fresh rate. Let's look at leaf coloration. At a lower temperature, the leaf redness of lattice was not affected by blue light intensity. But at a warmer temperature, as blue light intensity increased, the leaf redness of lattice increased by 76%. We also looked at the interaction between blue light intensity and air temperature in basils, but there was no interaction. So to summarize the interaction between blue foot and flux density and air temperature, the higher air temperature amplified the effects of a low blue foot and flux density on increasing lattice growth, but at the expense of crop quality. And blue photoflux density and air temperature did not interact to determine their effects on the growth and quality of basals. So I would like to conclude that the light spectrum could be fine-tuned considering air temperature and other environmental factors, as well as trade-offs that exist between biomass accumulation and crop quality. And interactions between the light spectrum and air temperature depend on plant species and were greater in lettuce than in basils. And I would like to thank their support and thanks for your kind attention.
All right. Thank you, Jiyong.
No problem.
Okay, so a few minutes for questions for Jiyong. How did you all measure plant redness to quantify the percent increase of percent increase or decrease from the treatments?
Basically, we used a top to down view photo to measure the redness, and we took the photo and removed the background and extracted some color values from the photos and used the redness value for our analysis.
Okay, so I have a question.
Sure.
So you tested red and far red, far red addition, right? Basil, I see the increase of dry mass. Did it dilute flavor of basil or it didn't?
Sorry, can you clarify the question again?
So I was wondering if far red leaf expansion of basil or biomass expansion would reduce the flavor intensity of basil leaves.
I didn't test the flavor contents of basils, but I agree. I guess if there is a accelerated growth or development, there should be some decrease in other sides of the plants, like flavor or things like that.
Did you actually taste?
No, I didn't.
Oh,
no,
you don't.
I didn't.
You don't. Okay, because it's all go to the analysis.
Oh yeah, for measuring
Okay.
dry weights.
Okay, of course. Yeah. Anyone has question? For Jiyong? Okay. For Devin and okay, so that we're gonna hold this because we're gonna have everyone come back. Anyone specific to Jiyong? Question? Okay, then let's you can stop sharing, Jiyong.
Okay.
And then okay. Welcome back. So thank you again for 3 presenters, excellent from Michigan State. So the question I like to cover here for Devin and all of the presenters, this is again question from Ed. I just purchased a package of rainbow Swiss chard seeds for my outdoor veggie garden. What lighting strategy would you suggest if grown indoors? Rainbow consists of green, red, yellow, and purple. Okay, so different pigments, different species. Just out of your, it's almost like an oral exam or some sort. Out of
That's what I was thinking.
out of your knowledge, scientific knowledge, or based on what you have done, or based on what you read, how would you approach? What lighting strategy would you suggest?
I think probably the first question, I have counter questions like, what lights do you have? Can you adjust the spectrum? Can you adjust the intensity? Is it like a cheap little like $20 Amazon light, or is it a light specifically for growing? All of that aside, if I could do anything and that wasn't a factor, I would probably start them out under maybe 150 to 200 micromoles of light, and then maybe hit them with a high intensity lighting a couple days before harvest to maybe really get those phenolics and those benefits of microgreens going. Personally, that's just my
Anyone else? wanted to jump into that. So
I
that-
would
Yeah.
mostly agree with that. I, since I like the light spectrum, would look at that, I guess. And if you want to enhance coloration and possibly nutritional value, think about incorporating blue light into the spectrum, especially if you're doing mostly red light, which a lot of those grow lights may be. But then again, also, if these are being grown in an area where you are frequently around, you may not want a red and blue spectrum, as it's not very pleasant to be under. So looking at white light supplemented with things like a little bit of far red and blue light could possibly be good for increasing growth and coloration a bit.
Maybe follow-up question. How significant are the differences in light spectrum requirements of various species of leafy greens?
It's a tough one to answer, I think. At least in my research, I've only grown lettuce, so we haven't done any studies that directly compare different species. But from reading plenty of papers, it seems like there are definitely some that are more responsive to changes in the light spectrum than others. And it can also depend on the light intensity and light spectrum combination, I think. So to find out, you really have to experiment on your own and see what the best combination may be for that specific leafy green, I think.
Yeah. Would you say leaf lettuce or red leaf lettuce actually more responding compared to other red leafy greens? Or
I
I
think
believe-
so, yeah.
Go ahead, Nathan, sorry.
I think so, at least under a lower intensity light like I've been using. If I don't include any blue light in the spectrum, a red leaf lettuce cultivar will look pretty much like a green leaf cultivar, and then adding even, say, 20 micromoles in a 180 total spectrum of blue light can really bring out some of that coloration, for instance. So yeah, I think lettuce is pretty responsive, at least the cultivars that we've used.
Yeah,
I
I think
believe that I've read that as well, that lettuce is particularly responsive to light quality.
Yeah.
Mhm.
yeah, and in my experience, lettuce is more sensitive to light quality than basil and kale. Yeah.
There we go.
So the rainbow chat that may not respond to light quality, huh?
It may just be an intensity thing.
Yeah. Yeah.
And because it is garden grown or sun grown plants, it may not respond quite that well to the light quality than to light intensity or things like that.
So the lutein content, so that carotenoid, right? You did, okay.
Yeah, I did look at lutein comment, and we had talked about including what I found as far as lutein and several other carotenoids and chlorophyll went in this presentation, but I personally just didn't feel ready to talk about it. Like, I had done enough background research. I didn't want to put my foot in my mouth, but stay tuned. We'll definitely be discussing lutein in the future.
Excellent. Because that's a good carotenoid contributing to human health out of whatever the leaf lettuce contains, if I remember. So that's good.
Yeah.
Anyone else want to ask those three presenters? Participation of young researchers of this quality is quite stimulating for those of us who have followed this process. Oh yeah, and then I'd like to thank to our USDA SCRI program, which provided this educational mission to educate graduate students. All right. Yeah, thank you. Anyone try their lettuce growing strategies on tomato? Ed, you ask quite interesting questions. Which have much, I don't think it has anthocyanin. You mean leaves or the fruit? Fruit doesn't have anthocyanin unless it is anthocyanin rich specific tomato.
I have not grown tomato.
Yeah.
It's a little more challenging to grow in our indoor farm setup. We tried a trial of like micro tomatoes before, and it's a bit messy sometimes with the fruit.
Yeah, so the tomato main fruit pigment is a lycopene, carotenoid, and then I think lycopene is also responding to light quality, but I don't think anyone did indoor study, as Nathan said. It's not easy material to grow in university, although I think in the future it might have more information coming. There might be more information coming out because the industry started testing tomato in the indoor farming setup. So the comment, I'm not sure. Eric's comment, is that maybe about tomato? Okay. Hard to grow indoors as you have to hand fertilize. Oh, you mean the pollination? Yeah. But bees are, I think, used also for tomato growing. All right. Anyone else specific to what presented, discussed? Oh, to Devin, there's a question. Did you also cool the water temperature for your experiment since you grew using a hydroponic system? What's the nutrient solution temperatures when you reduce the temperature?
We didn't cool the temperature or the nutrient solution, but it was cooled by the decreased air temperature. Usually within like the first day, it was pretty similar to the air temperature. Yeah,
tend to follow, right?
Right.
Yeah, tend to follow air temperature. So I was thinking, okay, it's running out of time, but I was thinking, is there a way to just cool the plant temperature without cooling air temperature?
That's something Roberto and I had discussed and maybe gonna work on in the future.
Great, because there's a radiative heating, so why not radiative cooling?
Right.
Yeah,
Right.
so anyway, I just thought about that. But anyway, it was excellent presentations. I really enjoyed research talks. And then I appreciate all of you, Jihon and Devin and Nathan, and then also your advisors who allowed you to speak. That is very important. And thank you very much, Roberto Lopez and Eric Ranco. Thank you. All right. Thank you so much, everyone, being with us. And then see you again next month. All right. Bye.